We are going to hear from a company that you may have heard their name, but you haven't heard from them in relation to their FOP work yet. So this is a really exciting opportunity to have them present to you today. And we're also very grateful to them for being one of the leadership partners for the family gathering. And with that, I'm pleased to introduce you to Insight Pharmaceuticals. And we're going to hear today from Heather Jones, Paul Smith, and Kurt Brown. So with that, I'm going to turn it over to you, Heather.
Hi, good afternoon and good evening. I'm happy to be joining you today, so thank you very much for inviting us to come speak. And on behalf of Insight, it's an honor and a pleasure. So I'm just going to move this. I'm sorry, I'm having the same problem. There we go. Apologies. Okay, so I'm going to read this slide to everybody just so we're all on the same page. And so I'll be speaking to you with my colleagues about our ALK2 compound. It's an ALK2 inhibitor also, and we refer to it as INCB000928 or 928 for short. And we just wanted to make mention that the efficacy and safety of this investigational compound has not been established just yet. There's also no guarantee that the compound will become commercially available. And right now, it's just an investigational product and not approved by the FDA or any other regulatory agency. So before I go on, I guess I should introduce myself. My name is Heather Jones, and I work in global medical affairs at Insight. A little bit about myself. I've been in the pharma industry for over 20 years, but my training and my background is in pediatric nursing. And I was a floor nurse at Children's Hospital Philadelphia, which I think a lot of folks are familiar with. And Dr. Kaplan practices adjacent to that hospital too, so from the same area. So I'm here to introduce Insight, and I thought we were the new kids on the block, but Caris is even newer than us. We were established back in 2002 when a group of 11 scientists left DuPont, and they started their own organization. And it's funny because those 11 scientists are actually some of them are still actively working in the company today. And we've been around and established now more than 18 years. We have about 1,600 employees, and we have a huge global footprint. So we encompass North America, Europe, Asia, and I'll go through some of those sites with everybody. And so the one thing that we're most proud about is our motto internally and externally is that we really follow the science. So we really stick with the scientific innovation, and we follow the biology and the chemistry. And we really look to discover first-in-class and best-in-class medicines for patients with unmet need. Oh, let me go back to the appropriate slide. Apologies. I'm tanking on the IT here, huh? Okay. So just a little bit of Insight's history over time. So like I mentioned, we were discovered or started in 2002. And then just a few short years later, the JAK2 mutation was discovered in the MPN space, which is oncology. It's myeloproliferative neoplasms. And then just 2 years later, we had the first investigational drug approval, an IND for ruxolitinib, which you'll see at the bottom is our Jakafi compound, and that was approved in 2011 for myelofibrosis. And then you can see over time the company had grown. We expanded into Europe. We expanded over into Tokyo. We became part of the S&P 500. We also shared in the success of Olumiant, which is baricitinib, and that's a compound that was developed by us and then sold over to Eli Lilly in fields like rheumatoid arthritis and some other inflammatory conditions. Ultimately, if you fast forward to around today, we have now an office in Canada. And we have multiple approved therapies, mainly in oncology and hematology oncology. Okay. All right. And so ultimately, we do have 7 products, and this is our global footprint. So we started off in Wilmington, Delaware, and that was that picture on the first slide, and we've now expanded. We're headquartered there still in the U.S., but we keep expanding, and we're looking to have a bigger global footprint, and we're hoping that we can ultimately bring our compounds to more of the globe. So that would be great for us and for the patients that we serve. This is just a snapshot of our portfolio. So we span targeted therapies, immuno-oncology. We have some partner compounds, but what we'll be talking about today is the inflammation and autoimmunity profile. And that's down at the bottom left in the pink. Just to take a deeper dive, what we have here is our later stage programs, which are a little bit more advanced. We have a topical JAK inhibitor, ruxolitinib cream for atopic dermatitis, and we're looking at it also in vitiligo. But we have an oral JAK1 compound that we're exploring in an HS disease space. But then you see over on the right-hand side some of our earlier programs, and FOP fits into that profile for us. So you can see here from just this small snapshot is that we have a strong focus on areas with no approved treatment but high unmet need. And this is our first foray into FOP, but we're very excited about it, and we feel very compassionate and compelled to make this work and hope that this brings a successful treatment option. And with that, I'm going to hand it over to my colleague, Paul Smith.
So hello everyone. So I'm a scientist. I'm not commercial. I'm not medical affairs. I have a research lab at Insight Corporation. We do a lot of the basic fundamental work internally. The other thing I'd like to thank the organizers is not only as a scientist, it's great to engage this community, but also I'm actually the parent of a small boy who has a spontaneous ultra rare genetic disease. And I can tell you that this kind of event is hugely important as a parent. So thank you very much for this opportunity. So we've heard earlier today about small changes in a gene resulting in a tiny change in a protein and how ultimately that becomes a very big biological difference. And this is an illustration of the ALK2 protein, a three-dimensional kind of rendering. And hopefully what you can see is there's a little tiny red kind of area that's highlighted. And this is ultimately the tiny change that occurs within the protein. But ultimately, of course, that has some big issues. And I'm going to talk about our compound INCB 928 and why we think that's going to potentially help families like yourselves. So this is a very small illustration. I'll try and walk you through it. So normally ALK2 signaling has a natural break. And this is a protein called FKB12, and it inhibits the activation and basically balances and helps control bone formation. Now, unfortunately, that small change in that protein ultimately means that that natural break can no longer work, and therefore all this downstream signaling, all these molecules that you see heading towards the nucleus remain active and biologically can drive bone formation. And this was going back to Dr. Kaplan's earlier presentation. This is the leaky faucet. This is the biology that underpins that drip of cellular activation, ultimately leading to ossification. Now, one of the reasons, and a couple of the other speakers have addressed it, clearly this is an overactivation of ALK2, and our molecule that we've designed, it's an oral small molecule inhibitor of ALK2. It's highly potent, and it's designed to basically inhibit that biology. Now, importantly, our molecule is selective. We've tested against the other ALK family members, and we get a very good selectivity. But actually, when we're designing a new molecule, we don't just look at the family members, we look broadly to see what other potential side effects there may be. We try to predict what the future may be. And what I can tell you is that 928 has been tested against over 500 proteins found in the human body. And what we can say is that as a pharmacologist, if I'm testing against 500 compounds, if I see a drug that's binding to lots of different proteins and causing lots of off-target unusual effects, we describe that molecule as being dirty. It worries us. We think there's going to be problems down the line. Now, the good news is when we run these tests, when we screen against over 500 proteins, the good news is that INCB 928 looks very clean. So this gives us encouragement that we've identified a very potent and selective molecule. And this, of course, is the ultimate goal when designing a drug. Now, obviously, we've designed a molecule, but we need to test and see how it works. And you've heard through many talks during today about the FOP mouse model, and it's a very powerful tool for us to evaluate potential new therapies. So I'm actually showing you some data that no one else in the world has seen before other than a handful of FOP doctors. This is kind of our first time we're showing these graphs, so and we're very happy to bring it to this family gathering. So the design of the study is very simple, is that we induce an injury in the muscle of the mouse, and then what we've allowed is a few days, about 3 days to pass, to let the biology, the inflammation, the bone process to just begin. And then we've started orally treating the mouse with our drug, and we're comparing that against a vehicle placebo group to see whether or not there's an effect. And we've dosed those animals up to 6 weeks of age, 6 weeks post-injury. And what you can see in the bottom left are some images. And they're images, they're CT scans taken from the vehicle placebo group over time, and underneath is the 9 to 8 group. And what you can see in the vehicle group is that at 2, 4, and 6 weeks, hopefully you can see it anyway, where the arrow is, there's bone formation that should not be there, and it's close to the knee. And actually 9 out of 9 animals in this particular experiment all shown bone formation at the site of injury. In contrast, when we've dosed those animals with 928, none of the animals out of 12 in this experiment showed any sign of bone formation based on the imaging results. Now, pretty pictures are very nice, but I'm a scientist. I like to have graphs. I like to do statistics and get to the hard numbers. And this is what's on the right-hand side. These are CT scans from the same animals in the experiments. And what you have here is the vehicle-treated group in the white bar showing that there's bone formation where there shouldn't be. And then we've tested 4 doses of our drug 928. At the very, very low dose, we've got a very tiny amount of bone, but it's statistically significant and it's over 90% inhibited. And at the 3 higher doses, there's no bone formation whatsoever. So on the scan data, it's looking really promising. But I mentioned that the injuries induced close to the knee. So one of the important things as well is, okay, is knee joint function still okay? We can't see much on the image, but is the knee still working? So we've looked at joint flexibility in these animals. And again, you can see the vehicle dosed mice have a clearly reduced knee flexibility. And at the 4 doses of 928, we're seeing basically a complete resolution of that problem. And I can tell you that that joint knee flexibility level of those 4 drugs is basically similar to healthy animals. So it's working not just on the imaging, but it's working in terms of absolute function. Now, in that first experiment, we dosed the mice after just 3 days of injury. So, we didn't give the biology, we didn't give the FOP biology much chance before we started using our therapy. So we did another experiment, and this is a much higher bar. This is harder to block. And the way we've designed this study is in the top left. Same thing, we're inducing the injury in the muscle to start the bone formation. But instead, we've waited over 2 weeks. We've literally done nothing. We've just let the biology, the ALK2 mutation driving the FOP, just let it have its time. And only after about 2.5 weeks have we then started to dose the mice orally with our drug or the placebo. And what you can see in the right-hand side this time is, again, some images from the groups, the vehicle versus the 928. Now, that first set of images is a baseline, so none of these mice have had any therapy yet. And you can already see that close to the knee where the arrows are, there's bone formation occurring. So there is already some bone formed and the disease is progressing. And then we've started the treatment, and we've got images from the 1 week and also 3 weeks post-treatment. And if we focus on the 3 week at the bottom right, you can clearly see in the vehicle treated group, there's a lot of bone formation that's occurred since the injury. And in fact, on the right hand side with the 928, there's a tiny little bit of bone still forming, but if you compare it against the baseline, it looks very similar visually. And again, as a scientist, I want to get some numbers on that, and I want to look across many animals. And here's on the bottom left-hand side is the in-life CT scan and also the end-of-study CT scan. And what you can see is that, there was some bone formation, but in the vehicle-treated animals, the open circle, the white circles, you see the disease just gets worse and worse with time. Whereas with 928 dosing, you see basically we've stabilized disease, there's no progression. So between the 2 experiments, we've shown that early intervention can stop an early lesion, an early ossification, but also in a pre-existing ongoing ossification process, we're able to stop its progression. And this gives us a lot of encouragement. And as I say, these are not necessarily easy hurdles for drugs to jump. I'll now pass you over to my colleague, Kurt Brown, who's leading our clinical team.
Thanks so much, Paul. Very exciting data, and it's really exciting that we're able to have the world premiere of that data here at the family gathering. By way of introduction, my name is Kurt Brown. I'm a pediatrician by training, actually a pediatric gastroenterologist. Worked for many years, over a decade, at the Children's Hospital of Philadelphia and was faculty at the University of Pennsylvania. And now I've been in industry for, 20 plus years now, developing drugs for both adult and pediatric patients. When I add those numbers together, I get a little nervous, but nevertheless, I'm so happy to be here today with you and share a little bit about our clinical development strategy. What is our molecule? You've heard that story from Paul. And now where are we heading? So just to refresh your memory, this is a molecule that's new to Insight. We in-licensed it or purchased it from Novartis in 2019. Since that time, we're doing many of the studies that you've heard about already. We're looking at this drug in healthy volunteers, first with just single doses, and now more recently with multiple doses, all in the effort to try and understand the safety and ultimately the pharmacokinetics, or how the drug moves through the body, so that we can pick an appropriate dose and begin our clinical trial in participants with FOP next summer. On this slide, I just wanted to establish that we are new. I think Michelle had said that. We're trying to be actually good citizens of the FOP community, and as such, we've kind of jumped in full force with our collaborators at the IFOPA. We're certainly here at the family gathering today, but there's a bunch of different initiatives that we think that are really important in establishing our credibility here. One, we definitely are anticipating support at the registry. We think that any data that comes out of our clinical trial should be shared liberally within the FOP community. And we've actually taken the opportunity to use the newly developed patient-reported outcome tool called the Prompt. We'll actually use that within our clinical trial in the coming year. We've been engaging many of the people that you've known, seen, and talked about here today. And I think importantly, we've been talking with the IFOPA not only at an organization level, but at an individual level. We've been graciously admitted to the homes and had conversations with multiple families and patients. And I think it's really critical for us to understand as well as we can this patient population, so that we can create a clinical trial that's reasonable to participate in, knowing the constraints of some of the health issues and the geographic issues going on with this population. So in the next slide, we have our clinical trial at a very high level. These are the bones of the clinical trial that we're trying to establish now. Again, the idea is to have this trial launched in the summer next year. To us, summer means June, July, August timeframe. We think that once the trial is initiated, we can recruit it, meaning getting enough patients to come into the trial over an 18-month period of time. It is a global trial, so we'll be looking literally almost at most of the countries in the world for an opportunity. We haven't landed on any specific country yet at the current time. The objective is to use the ALK2 molecule 928 to prevent further heterotopic ossification, our number one goal. But certainly we will be looking at a whole host of other issues, such as, can we diminish the number or severity of flares? Can we alter the symptoms of FOP? And I think somewhat novel, in a novel way, we think that this first trial will predominantly be adult patients, but we seek to actually include some adolescent patients within the 40 participants that we look to enroll. The trial design is very, very similar to what you've heard from other sponsors at this point. You've heard about a placebo control period, a period in which the patient or the investigator does not know whether they're actually receiving the real drug 928 or a mimic, a placebo. And that part will take place for about 6-month period of time. The intention here is to gain data. What does this drug look like? What is its safety profile? What does its efficacy look like? Once that 6-month period is completed, then we would invite all the participants to come in a open label, at least 12-month extension, meaning everyone would get drug at that time. The other things that we're contemplating, and actually we're looking for feedback from this community on, are some options that would make this drug, this clinical trial, attractive. One of the things is a flexible oral administration. At the current time, we think that our drug will be a capsule once per day. We're certainly making it so that you can actually break the capsule open and disperse a whole bunch of small little fragments that are about 2 millimeters in size. So those 2 millimeter size particles could actually be suspended in a liquid, water, juice, or put on yogurt, ice cream, whatever vehicle you want, in order to facilitate the ingestion of our product on a daily basis. The other thing that we're trying to understand is, can we minimize some of the invasive procedures that happen around clinical trials? There's no getting away from the fact that phlebotomy, taking some blood, is necessary. But we've come up with actually some kind of interesting ways. We found out that our drug circulates through the body, and actually if you take a sample of saliva for someone, their spit, and you look at it, it very, very closely parallels the concentration of the drug that we see in the blood. So we look to actually substitute some of the phlebotomy, some of the blood draws that would normally be done to understand the circulation of the drug, and just have the opportunity to collect spit from our participants. So we hope that that's attractive. Lastly, we're in a pandemic. No one can predict the future. We are all crossed fingers at this time, thinking that a vaccine will come and hopefully modify our current situation. But even in anticipation of that happening well or not happening well, we're looking ways to minimize travel. Minimize travel means patients would definitely need to come to the investigative site for some key procedures. But we think there's an opportunity to have the study come to the patients. That means coming to your home, should you wish, perhaps coming to a regional center, a healthcare center that's local to your environment, rather than the need for someone to get on some kind of vehicle and travel great distance, committing great effort to do so. So that's just a very, very high level view of our clinical trial. On the last slide here, we're looking for your input. We have a website, we have email address, we have a phone number. These are all ways to collect information. And should you want to comment on anything that you've heard today outside the family gathering, for instance, please feel free to contact us. We'd be more than happy to have your insight. With that, I say thank you so much for the opportunity today. It's been wonderful.